Hello, everyone, and welcome to today's The Scientist webinar. I'm Tiffany Garbett, Assistant Science Editor for The Scientist, and I'll be moderating our discussion. Today, our speakers, Dr. Chris Heger and Dr. Johan Skog, will discuss exosomes, some of the latest diagnostic applications in the exosome field, and introduce simple Western technology. We like our webinars to be interactive. We encourage you to send us your questions or comments at any point during the webinar, and the speakers will address these during the Q&A session following the presentations. To ask a question, simply click on the Q&A tab and type your query into the question box located on the right side of your screen. We will try to address as many of these questions as we can during the Q&A session. The webinar will be archived on the Scientist website, and we will send you the link via email within a couple of days. Please note that you will not be able to download the presentation slides. I'd like to take this opportunity to thank our webinar sponsors, Protein Simple and Exosome Diagnostics. Protein Simple develops and commercializes proprietary systems and consumables for protein analysis that ultimately help reveal new insights into the true nature of proteins. Their goal is to make protein analysis simpler, more quantitative, and affordable. Their comprehensive portfolio of tools includes simple Western systems that quantify protein expression and biologic systems that probe the structure and purity of protein-based therapeutics. Exosome Diagnostics is a world leader in developing liquid biopsy-based diagnostics and empowers the world's leading biopharma companies' biomarker discovery, translational research, and clinical trials through partnerships. Exosome DX offers services using RNA, cell-free DNA, and proteins with their proprietary technology to explore and identify biomarkers. It is the first and only company that can simultaneously isolate and analyze exosomal RNA and circulating free DNA in a single step to achieve highest sensitivity for detecting rare mutations. Exosome Diagnostics is a core provider to support advancement of patient care. Our sponsors have provided us with some helpful resources related to today's webinar, and we have posted these in our Handouts tab located on the right side of your screen. You can access and download these documents at any time during the webinar. And with that, let me introduce our first speaker, Dr. Chris Heger. Dr. Heger is the Senior Manager of Application Science for the Analytical Solutions Division of Bio-Techne. He leads a team chartered with creating scientific collateral, fostering collaborations, training, and coming up with new applications. Chris has expertise in immunoassays, capillary electrophoresis, antibody design and production, and chromatography. In recent years, his team has focused on developing analytical solutions for cell and gene therapy researchers. Chris received his doctoral degree in pharmacology from Cornell University and completed his postdoctoral training at the National Cancer Institute. Working in the Antibody and Protein Purification Unit, Dr. Heger oversaw antibody development projects from immunogen design to final product. A unique aspect of his postdoctoral training was his role in the evaluation of new technologies, serving as the gateway for cutting-edge instrumentation to get into use at the NCI-NIH. Through his role, Chris became well acquainted with Protein Simple, and after completing his postdoctoral training, began working for Protein Simple, which is now part of Bio-Techne. Dr. Heger, let's just take a minute to make sure your slides are up and running, and it looks like they are. Please feel free to take it away.
Hi, thank you for that great introduction, and thank you all for your attendance at today's webinar. I'm Chris Heger, as Tiffany mentioned, and I'm going to speak first about the advancements in exosome research tools and diagnostics. So in addition to our Exosome Diagnostics brand, which of course we're here to talk about exosomes, so you expect to hear from Exosome Diagnostics, but Bio-Techne also brings together several other prestigious brands, including Advanced Cell Diagnostics, Novus Biologicals, Protein Simple, Tocris, and R&D Systems. And all these brands are brought together to support your exosome work. Together, these brands provide a broad portfolio of tools for exosome research, including cytokines and growth factors, as well as small molecules, and media and supplements. And a lot of these various reagents are available at GMP grade as well. We also offer world-class immunoassays and ISH technology, and a wide and diverse array of proteins and antibodies for your work. In addition, as we'll talk more today, Protein Simple provides analytical instrumentation, and finally, Exosome DX for cutting-edge exosome isolation and characterization. Our goal is to offer all of these innovative solutions with flexibility and customization to fit your needs with the consistency, reproducibility, and scalability you require. And today, I'm here to introduce one of our protein simple platforms, Simple Western. And later, Johan is going to talk about its application onto exosomes. Analytical tools are constantly evolving to adapt to the rapidly changing research and development landscape. Sometimes these changes are geared towards allowing the researcher to make faster insights, as is generally the case when using a smartphone today compared to a desktop computer from 20 years ago. And sometimes these changes are meant to help provide a better view, as has been the case of the evolution of the microscope. And when I think about biotechny in the context of these pictures and exosomes, I confidently can put both Protein Simple and Exosome DX alongside these images, as these brands are helping make faster insights and provide a better overall view and understanding of exosomes. The traditional Western blot has been around for 40 years, and while being a key research tool, still persists in a form that looks pretty similar to what was available 40 years ago. There have certainly been innovations to help improve the overall process, but it's still just that, a process. It's a process that requires a lot of gear and a lot of time, and it yields results that aren't often worth the effort. The Simple Western platform, on the other hand, takes the entire process, innovates, and automates it, all in one box, completed start to finish in as little as 3 hours. Now, personally, having run thousands of Western blots the old way, I truly wish I had had this platform around when I was doing all that work years ago. Now let's take a quick look at how the Simple Western platform works. Simple Western immunoassays take place in a capillary. Your sample, primary and secondary antibodies, as well as separation and running buffers, are loaded automatically from a specially designed plate. Jess starts by aspirating the separation matrix, followed by the stacking matrix into each capillary. The stacking and separation matrix mimic your discontinuous STS page. Next, your protein sample is loaded into the capillaries. And finally, the capillaries are lowered to make contact with the running buffer, and voltage is applied to separate the proteins by molecular weight. Once the separation is complete, UV light immobilizes the proteins to the capillary wall. With the proteins immobilized and separation materials cleared from the capillaries, Jess starts the immunoprobing process. Samples are first incubated with primary antibody, and then, in the case of chemiluminescence, with an HRP conjugated secondary antibody, which recognizes your primary, and finally a chemiluminescent substrate. The chemiluminescent reaction is recorded by a CCD camera in a series of images over time. Automating and moving over to Simple Western ensures that you get the same answer every time. While traditional Western blots can typically yield replicates with CVs between 20 and 35%, Simple Western is going to do considerably better for you. Shown here is an example within a single run where we looked at a multiplex of PI3 kinase and CDK6 across 24 replicates. And the resulting CVs from PI3 kinase and CDK6 were 4.4 and 9.1% respectively, considerably outperforming what you would get on a traditional Western. And of course, it's nice to have reproducibility within your run, but across days is also extremely important. And to illustrate that, we had 4 different users across 4 different days run the exact same experiment. And plotted is their average signal for their replicates. And you can see overall, the 4 users had pretty consistent data. In addition, within their own data sets, the CVs were all below 12% for 11 replicates each. And at the end of the week, we averaged all their results together and got a CV for 44 replicates under 10%. So these data show that the Simple Western platform will give you reproducible results either within your run or day to day, giving you more data confidence. Additionally, the Simple Western platform can provide quantitative measurements for your target of interest. All you need to do is include a recombinant protein or a purified protein as your reference standard, and the software will take care of the rest. So first, let's look at a quick example of absolute quantification of endogenous ERK2 protein. The researchers in this experiment used a GST-tagged ERK2 protein and spiked that in a titration in a constant amount of lysate. The entire capillary was probed with a pan-ERK1/2 antibody, which detects both the recombinant ERK2 as well as the endogenous proteins. The recombinant protein was then used to make a linear regression, which could then be used to back calculate the amount of endogenous ERK2 in the sample. This is one example of how our true quantitation can enable measurements of the endogenous proteins. But I would say that this technique is also used by biopharma. And here's an example where a particular customer is looking at a biopharmaceutical protein product or drug substance in a titration. And there are actually two protein products for this drug substance, and both can be quantified either independently or together to make a really nice, tight-fitting linear regression. The last feature I want to touch upon with Simple Western that really pushes it apart from traditional Western is the dynamic range and sensitivity. And to illustrate this, I'm going to show you a graph with a HEK293 lysate titration on it, where we're probing for GSK3 alpha. But first, let's look at the traditional Western blot results. The traditional Western is able to detect GSK3 alpha anywhere from about 10 megamil down to a little bit above 0.01 megamil, providing a little bit over 2.5 log dynamic range, as you would expect. When we ran the same set of samples on the Simple Western assay, we actually had to titrate the sample a lot further because the Simple Western platform was so much more sensitive. So in addition to having ability to detect the same high end of the curve, we're able to detect several logs of magnitude more sensitive for GSK3 alpha in this lysate. So in addition to the high reproducibility, the true quantitative nature of Simple Western, you also have an extended, longer, wider dynamic range with exquisite sensitivity. We have an entire family of Simple Western instruments, including the Jess that I showed you earlier, and Wes. And both Wes and Jess can run up to 24 samples in 3 hours. Wes and Jess are very similar, except Jess, being Wes's younger sister, actually has a few new features, including dual color fluorescence, blot imaging, and reflex. We also have 3 instruments that can run up to 96 samples in a run, including the Sally Sue, the Peggy Sue, and the NanoPro 1000. The Peggy Sue in the middle is essentially a hybrid that combines the high throughput size run capabilities of the Sally Sue and the high throughput IEF runs of the NanoPro 1000 all in one box. All 3 of these instruments can complete runs in under 14 hours for up to 96 samples, and all feature our ultra-sensitive chemiluminescent detection. I've been a part of Bio-Techne for almost 10 years, and I've seen the entire adoption cycle of Simple Western platform, even starting as a customer beta testing instruments myself. Over that time, I've seen the Simple Western platform applied to a diverse array of samples, everything from crude cell lysates all the way through to pure proteins. These could be cell lysates, mammalian or Arabidopsis origin. I've seen tissue extracts from mouse corneas and skin lysates and so on. Biofluids from serum, plasma, urine, CSF fluid, recombinant proteins, monoclonal antibodies, viruses, and as you're going to hear more about today, exosomes as well. The point there is, if you have a sample, we have the ability to help detect it. And over the last 10 years, the Simple Western platform has also supported over 900 peer-reviewed journal articles and patents. And rather than looking through all 900 publications here today, I figured I'd make some word clouds to help illustrate their impact. And I did this first by research area. And when I did this, you can see some really powerful words coming out here across early discovery. You see signaling, drugs, cell, and across later stage processes as well, like bioprocessing. You see methods and optimization there. I also made a word cloud looking by journal title inclusion, and some more powerful words come up here as well, including research, the Nature being one of the top journals, USPTO, the Patents Office, Cancer, Medicine, Physiology, and Reports. So Simple Western is used for a wide, diverse array of samples and has been published and realized for its value in a wide variety of publications. Just launched, I'd like to spend a few minutes talking about our newest feature for Simple Western called Replex. Replex is an automated way to reinterrogate the same capillary post-separation with another round of probing and detection. You can think of it like the best of a stripping and reprobing without the harsh chemicals or poor reproducibility. So let's look at some of that data. So in the experiment I'm showing in this top panel here, we're looking at dual detection of phospho-AKT, which will be in the chemiluminescent channel, and pan-AKT in an IR channel. We're looking at MCF7 lysates in either the absence or presence of IGF-1 stimulation. Now, in the control MCF7 lanes, we don't see any phosphorylated AKT, which would be in the black. We only see the pan-AKT shown in red here. However, upon stimulation with IGF-1, we see a rapid change, and we see phosphorylation of 473. We see a perfect overlay of the phospho and total here. And then same thing for the threonine 308 is also phosphorylated when detected with that specific antibody. Now, to make sure that the changes seen here are real, you want to make sure that the amount of protein loaded between the capillaries is the same. You know, and certainly the classical way to do this would be to use a loading control like GAPTH or Actin. But if you really want a better view of what's going on in that sample, you should really look at the total protein. And so enter Replex here, where we're going to essentially reuse the capillaries. We're going to replex them and then reprobe them. In this case, we reprobed for our total protein. And so here are those 4 same capillaries and the total protein landscape, as I like to call it, what's going on across the entire separation range in those capillaries. And our Compass software is able to take the total protein area as well as the immunoassay area under the curve, shown here, and result in what's called a corrected area here, which takes into account this total protein area and applies it to your immunoassay data. We are really excited to have this feature available, and we're very excited to see how our customers have already started using it. I wanted to spend a few minutes briefly talking about Peggy Sue and what she brings to the table. Our higher throughput instruments, including Peggy Sue shown here, can really help you get the most out of a precious sample. So let's walk through a quick example so I can show you what I mean. Let's imagine that these 8 individual wells that I've drawn here are from our Peggy Sue plate. And we're going to fill them with patient samples. We've got 2 patient samples here. And we're going to look at both whole cell and nuclear extracts. In addition, those 2 patient samples each have 2 treatment conditions, either in the absence or presence of TNF-alpha. Now, for each one of these sample wells, we're only going to put 5 microliters of sample into the well, and that's it for each sample. Now, the beauty of Peggy Sue is that each one of these 5 microliter wells can be interrogated multiple times by different capillaries and probed with different primary and secondary antibodies. And what this allows you to do is take that very precious 5 microliter sample and probe it multiple times with different antibodies, as shown here. And what we're doing here is looking at the entire NF-kappa B pathway across these samples. And you can see some really nice changes where the NF-kappa B P65 largely doesn't change in the whole cell. It does increase in nuclear extracts upon stimulation. IKB goes down or decreases in both patients in the nuclear, upon stimulation. And CREL has essentially the opposite trend. So this is a lot of information that you can gain off of essentially single samples if you go down at one column here. So that same 5 microliters, which would give you 1 well essentially on a traditional Western, can give you 8 times as much data when you're using a Peggy Sue instrument. In addition to the size separation capabilities I've described on both the Jess as well as the PEGI-SUE, we also offer charge separation on the PEGI-SUE. In this separation mode, we use the same capillary capture and in-capillary immunoassay to leverage the isoelectric point of the protein. This allows one to look at the subtle changes in a protein that cannot be detected by size separation, for example, like phosphorylation, which only adds 80 Daltons to the protein. With charge separation, and the example is phosphorylation, each phosphorylation event acidifies the protein, moving it to the left on this PI scale. Let's look at an example for the MAP kinases ERK1 and ERK2. The beauty of this system, in part, is the ability to use a single antibody to characterize post-translational modification. This is quite beneficial if an antibody for your specific PTM doesn't exist or isn't readily available. So when we look at helilysates for ERK1/2 probed with a pan-ERK1/2 antibody, we see essentially two peaks, one for ERK1 and one for ERK2. And we can quantify the amount of unphosphorylated versus phosphorylated ERK species here. Upon stimulation with EGF, we see a rapid increase in both the diphospho ERK1 and ERK2 species, as well as the concordant decrease in the non-phosphorylated forms of the protein. These phosphoisoforms can be easily quantified and show that upon stimulation, the ERK1 and ERK2 proteins are almost 90% phosphorylated. This exact assay system has been also used by researchers and published where the use of the charge assay has been used to look at ERK phosphorylation in patient samples. And specifically, I show an example here looking at phosphorylation in lung cancer and emphysema. In the top panel are the ERK signatures for normal lung tissue, shown in blue, and from lung cancer tissue, shown in red. Now, to me, there's essentially no visible difference between them, and predominantly we see ERK1 and ERK2 only. However, in the bottom panel, where the normal lung tissue is compared to lung tissue from emphysema patients, there's a stark difference, and we see that the assay might be useful to better understand this disease. These observations are quantifiable, and the data were shown to be significantly significant. The charge separation has been used in many other assays as well to look at other post-translational modifications, or in other cases where proteins can't be separated by size. I tried to summarize some of the key reasons and benefits to use the Simple Western platform if I haven't already convinced you. If you need faster time to answers and you want less hands-on time, the fact that we can run a complete start to finish assay in 3 hours with as little as 30 minutes of prep time is quite attractive. In these times, if you've been out of the lab for a bit and now you have a backlog of samples and need more throughput, again, being able to run a really fast, high throughput instrument like this is going to be beneficial to you. If your samples are very valuable, very precious, either coming from a patient or going through a very strenuous extraction protocol, using only 3 microliters of a dilute sample on our platforms is going to be very beneficial and allow you to stretch that precious sample further. And to that end, if you need multiple answers out of that same sample, using PEGI-SUE or Sally-SUE or the NanoPro 1000, as I illustrated earlier, will allow you to interrogate that sample multiple times. If other immunoassays aren't really fast enough for you and you still require quantitation, I think running Jess or West in 3 hours is really going to be the answer that you're looking for. And certainly with the reproducibility and quantitative nature of the platform, the accuracy is going to be superior to what you would get on traditional Western or other immunoassay platforms. And if you need more than just size data, but you still need to quantify, the charge separation that we offer is quite unique and novel and not available on other platforms. And lastly, in these challenging times where most of us have had some restrictions on our lab duties or access, the ability to minimize your lab time, as well as access your data remotely when the run is finished, is certainly an attractive feature of the Simple Western platform. And building on these, I added just a few other key benefits to consider. And the first is that the Simple Western platform is scalable and it can grow to support your needs. It's supported by a long supply chain with consumables as well as for additional instrumentation. And all of our instruments have a small footprint, which allows for multiple units to be placed in a small space. These methods can be easily transferred to other sites, organizations, ensuring that you can scale and get the consistency across your different sites. All of our data for Simple Western is digital. So all the data, the records, the run files, they're all digital. That allows for easy access, easy analysis offline, easy data transfer between colleagues and sites, as well as easy data archiving. And the Simple Western software is available in a CFR 21 Part 11 compliant format for all your regulatory and auditing needs. And lastly, something that you certainly won't get with traditional Western is global support. The Simple Western product family, like everything in Bio-Techne, is supported by a global team. This includes global tech support services. We'll answer your calls at any time of the day. Your field application scientists will train you and help with your assay development. Your field service engineers to help install your instruments, as well as perform any of the IQ, OQ, PM, RQs, and maintenance service plans that you engage with us in. And with that, I'd like to thank you for your time today, and I'm happy to pass the microphone over to Johan.
Thank you, Dr. Heger. As a reminder to our audience, you may ask questions at any time using the Q&A tab located on the right side of your screen. Our panel will have a chance to address your questions after our next speaker, and that next speaker is Dr. Johan Skog. Dr. Skog is the Chief Scientific Officer and founding scientist of Exosome Diagnostics, now a Bio-Techne brand. He pioneered discoveries about exosomes and other microvesicles and their vital role, both as cell messengers and disease proliferators. Skog has a PhD in gene therapy and virology and a master's in biomedicine from Umeå University in Sweden. He later moved to Massachusetts General Hospital / Harvard Medical School to study tumor stem cells, during which he observed exosome vessels and became the first to show that tumor-derived mutations can be detected on exosome RNA from serum and other biofluids. These findings were published in a seminal paper in 2008. He later showed how these vesicles can be used therapeutically by incorporating gene therapy vectors into microvesicles as a stealth vector with change tropisms. Skog is the inventor of several novel exosome isolation platforms that are used clinically, see GMP manufactured, as well as exosome isolation platforms that were licensed to QIAGEN. Skog has greater than 138 applications, divisionals, or granted patents in the exosome field, spanning more than 35 patent families. He continues to expand the field of exosome biology and leads critical advancements in diagnostics, including the first exosome-based diagnostics tests that are now helping clinicians and patients. Dr. Skog, let's just take a minute to make sure that your slides are up and running.
Okay, can you hear me okay and see my slide?
We can hear you okay. Can you screen share your slide and let's make sure that we can see them?
I think I am. I hope you can see it.
No, we cannot see it as yet. Let's see. Can you stop sharing and try to reshare your screen again?
I will try.
Thank you so much.
Sorry about that.
Not a problem. And they're loading now.
Okay, excellent. Thank you so much for that introduction, and thank you everyone for joining this webinar. So I will talk today about using exosomes as a liquid biopsy, and I will talk about how some of these platforms that Chris Hager was just talking about can enable exosome-based research and also help you deal with these precious samples that you often have in these biomarker studies using exosomes. So to set the stage, I just want to sort of frame what exosomes are. So exosomes are these small vesicles released from cells into the surrounding. And when they are released from the cell, they end up in biofluids, such as blood, plasma, cerebrospinal fluid, or urine, and even saliva. And these vesicles are loaded with interesting material from the donor cell that can be used for biomarkers, as well as basic cell biology research and understanding this intercellular communication that happens to these vesicles. So in the middle picture here is a very nice illustration of two tumor cells growing in a body, and you can see this cloud of vesicles being released from them. And a single tumor cell can net release over 20,000 of these exosomes into the surrounding in any given day. And these vesicles, if you zoom in, you can see that they're loaded with material, so proteins, lipids, RNA. And tumor cells use these exosomes to its advantage. So they're actually part of the actual disease process, where tumor cells release these to stimulate tumor cell growth, blunt the immune response, stimulate angiogenesis, and many other things. So that makes them even more attractive as biomarkers because they're really part of that disease process. When RNA and other molecules are incorporated inside the vesicle, they become very stable. So that's also one of the key sort of hallmarks of a good biomarker. And it's a distinct entity of liquid biopsy that is very different than cell-free tumor DNA and CTCs and cell-free RNA. So why has so many people started to study these exosomes? Well, they're really a multifaceted molecule that can play a role in cancer immunosuppression, for example, where it's been shown that PD-L1 and cancer-derived exosomes can affect efficiency of immunotherapy. They can be used to modify the tumor microenvironment, etc. But they play a role also in other biological processes. For example, it was recently shown that exosomes are part of memory and learning, where, for example, the ARC mRNA are encapsulated by ARC, like in a virus particle, and transported over to the target cell. And this is a critical component of formation of these synapses and memory. They're also part of proteinopathies. So prion proteins, GRAPE, for example, is selectively targeted to exosomes. And in infectious diseases, it's been shown that tick-derived exosomes can suppress inflammatory responses and transmit flaviviruses. And a wide range of viruses can actually be incorporated inside these exosomes as a cell cloaking and basically protect the cargo so that the viruses are not seen by the immune response. And it also changes the tropism and the stability of the virus particles. So there's a lot of really interesting biological processes to study with these exosomes. And that's where some of these tools that Chris talked about come into play. When we talk about the biomarkers specifically, liquid biopsy is really booming right now because we're moving away from this morphological diagnosis of looking at a tissue slide to more utilizing a molecular diagnosis where we study either DNA or RNA to stratify the patients. And when you do that, heterogeneity is really a big issue. So when you take a tissue sample, you have heterogeneity both in space and time. So tumor cells, for example, is known to evolve very quickly. You can change the dominant genotype of a tumor in as little as 4 to 5 months when you apply treatment to the patient. So being able to track these changes over time and also monitor multiple lesions at the same time is a very critical feature of liquid biopsy and gives you real-time insight into what's going on. It's also been very exciting in the last several years because exosomes can also help the therapeutic development. For example, in immunotherapy or checkpoint inhibitors, we know that exosomes are released in every stage of the immunotherapy cycle, basically. So you have exosomes released at every stage. And when you add the function that you can use these blood draws or urine collections over time, you can develop even more powerful signatures using biomarkers that are not useful in tissue. Because as you can imagine, patients, we're all different. There's a huge heterogeneity in the human population of a variety of different molecules. And if there is heterogeneity and variability in the baseline level of a molecule, it's very hard to find a biomarker that works across the board at a single time point. But what liquid biopsy enables is that you can take multiple samples from the same individual over time, so that even if one individual has a higher baseline level of a biomarker, you can track if it goes up or down over time, and therefore utilize biomarkers that are not useful in a single point of time tissue sample. So when it comes to processing of biomarkers, there's really been a little bit of a crisis in the way we develop these biomarkers, because most biomarkers that are developed in literature fail. And this is not an exosome biomarker specific phenomena. This is a graph that I show from any type of biomarker that's been published. So you can see that there's over 6,000 papers on biomarkers every year, and there's over 1,000 patents on biomarkers every year. Despite that, there are very few of these biomarkers that are actually moving all the way to clinical use. And most of that is because these biomarkers, once they're taken into the clinic and run appropriately, the biomarkers fail. So it's very important to control for pre-analytical variables, probe compliant sample collections, and have an appropriate design in your studies. But more importantly, we need properly evaluated platforms that can consistently process samples with little variation. So we need to make sure that whatever we use to isolate the biomarker, it's unaffected by the biofluid matrix effects that can be very variable between individuals. And all of these processes, unfortunately, need to be optimized and validated for the particular biomarker of choice. When people are talking about exosome isolation platforms in general, there's no one size fits all, because every method have pros and cons. And some of this depends on what type of biomarker you're interested in looking at. Is it an RNA? Is it a protein? Is it a sugar, a metabolite? Is the biomarker present both in vesicles and outside of vesicles? Is it associated with a membrane or inside the vesicles? All of these things need to be taken into consideration when you choose your isolation platform. You don't need to have a platform that necessarily isolates something that is very well defined. It just needs to be very consistent. And this is an example of a clinical exosome isolation platform for exosomal RNA that we use here at Exosome Diagnostics. And when you look at these platforms, we always choose the most appropriate platform for the biomarker. So here's just a list of different types of isolation platforms that we've developed, where the Exsolution portfolio here are clinical grade exosome isolations that we use for our clinical studies, as well as our tests that are commercially available. We also have an example here, the ExoRNeasy, which is a research grade or MBA grade isolation kit. The UPrep kit is used for urine exosomes. Exsolution Eddy is utilized if you need to enrich certain vesicle types. So you enrich them based on a surface marker. So you do either enrichment or depletion. The Exsolution RNA kit is used whenever we need to profile RNA, and here we can profile over 30,000 different RNA targets in a biofluid sample. Exsolution Plus is for mutation detection, and Exsolution HT is for high throughput applications. Often when we talk about exosome isolation platforms, you hear that some people think that the exosome isolation is a complicated, cumbersome process and it's not standardized. However, we've proven over the last 10 years that that is not the case. The exosome isolation can be very easily standardized, and here's an example of a real-world reproducibility, basically, where I pulled 440 replicate extraction controls across 5 different urine batches from our CLIA lab. So these are real-world data that represent 440 clinical batches, and that represents over, tens of thousands of clinical commercial samples. And as you can see, the reproducibility of these urine extraction controls are extremely consistent. If you zoom in on just batch 5 here, you can see the CT values have a standard deviation of about 0.4 CTs across all these extractions, and that these probably involve half a dozen different CLIA operators and over a long time. So the Protein Simple products can help you in the standardization and in the way you characterize the exosomes that you isolate. And I want to show you some examples how you can utilize, for example, the JESS instrument for exosome characterization. So this is a semi-automated analysis of exosomal proteins, and it can be used from a variety of different biofluids. So the good thing with this platform is the high sensitivity. So it requires only minimal input requirements. This has been a sore point for exosomal researchers, because once they isolate their precious exosome cargo, they can sometimes use 80 or 90% of that sample for characterization, and very little is then left for the actual analyte readout. So in this case, we use the ExoEasy column, which is a very simple bind, wash, elute method for exosome isolation. So from a milloplasma or whatever input you want to use, you get about 250 microliter of isolated vesicles that you can use for RNA profiling, electron microscopy, functional testing, NTA, etc. And a very tiny amount of this volume can be put into the JESS for characterization. And here we show you some example of exosomal markers like CD9, flotillin, and HDL markers, non-exosomal markers like APO, A1, that people are often using as controls. And when we've done this, so we did this over 2 separate days using 18 separate isolations of plasma, and we found that we have a very robust CV readout of both CD9 and flotillin, about 6% CV on repeat measurements, 20% CV on repeat isolation, and with different operators, days, and isolation methods, about 30% CV. So that's really, really tight. When you're looking at different markers, you may need to adjust the stringency to increase your specificity for EV characterization. And here's an example of that, where if you utilize a plasma sample and you wash the sample once before you elute it, you can see that there's still a lot of albumin present in the sample. Note here the differences in the elute dilution. So this tells you how little material you actually need to see your sample. So you dilute your elute 1 to 10, and you only need a few microliters of this to detect the actual markers. From a urine sample, you may also need a little bit more washes, depending on what marker and what background you're trying to remove. So this is an example where you can see the albumin amount here. The 1 in 20 dilution from urine is because there's much less albumin present in urine, which it should be. The plasma had to be, exosomal sample need to be diluted 1 in 1,000. So we've optimized sort of the workflow and tested a variety of different protein targets that are commonly analyzed on exosomes. In this case, these are the MySev category, or the MySev recommended markers. For example, EV proteins, you have CD63, CD9, ALEX, annexins, etc. And as well as non-EV markers as controls, albumin, ApoA1 or ApoE. So here you can see what sort of antibodies you can use for these on the JESS system. When you're doing your experiment, it's always nice and reassuring to show that you have a dose response curve, right? And here we show an example of flotillin, where we go from 0.25 milliliters of plasma up to 8 milliliters of plasma. And you can see that the IgG background here is fairly constant. However, the amount of the specific flotillin increases with the higher volume. And this is shown through this electropherogram. You can also display this on a more classical Western blot way. And here is an example where from the same sample you can look at all of these markers. You can look at CD63, CD9, ALEX, annexins, flotillin, beta-actin, etc. So it's a very powerful system to use very little material from your precious sample and still be able to process a lot of different markers. Here again is another example of looking at what's in the total plasma sample if you load raw plasma on your device. And as you can see, if you look at plasma alone, you don't see the marker. So if you use the raw material, you can't see the marker. But if you enrich the exosomes by the isolation, you can get over a 600-fold concentration factor and start seeing these EV proteins. And the same is true for exosomes. So you can see exosomal markers from as little as 1 milliliters, 1.25 milliliters of urine. So why is this important? Why is there such a huge boom in exosomal research and analyzing exosomal RNAs biomarkers? Well, the advantage of these exosomes is that these liquid biopsies can go far beyond just looking at mutations and methylations that you do on cell-free DNA. The exosomal RNA allows you to do mapping of pathway analyses because the entire RNA transcriptome can be found. You can do splice variant analysis as well as improved mutation detection sensitivity. And if you use proper methods, you can enable complete RNA profiling from virtually any biofluid. So we've shown that you can do complete RNA profiling from plasma, serum, cerebrospinal fluid, urine, even saliva. And your library prep can be optimized to do either a whole transcriptome profiling or the exome or an exome plus link RNA. You can customize this as you want, and then do differential expression, look at mutations, insertions, deletions, fusions. And we've seen that when you do this, you get the same diversity and detection as you get in a tissue sample. So from as little as half a mil of plasma, we can see over 30,000 of the 56,000 genes and about 17,000 of the 20,000 mRNAs that are in GenCode. In every sample, you also include controls, so you can show that you can detect reproducibly down to single-digit molecules of RNA with a very high, very wide dynamic range to over 1 million copies of some of the RNA targets. And since the exosomal RNA is so stable, we've been able to show that complete exosomal RNA transcriptome profiling can be achieved from samples stored for over 25 years in biobanks, and that's quite powerful. When you look at the RNA content in an exosome, therefore you can successfully monitor the protein coding RNA. Here we were able to suppress the ribosomal RNA because most people don't want to spend their sequencing efforts on ribosomal RNA. You want to really see what the RNA targets that mean something for your pathway analysis. And you can do this from any biofluid. However, the RNA targets that you do see is very different depending on what biofluid you choose. So you can see that the RNA profile from CSF is different from plasma, and that's different from urine. And that's because these biofluids take up very different types of exosomes. So in urine, you have exosomes coming from the urogenital tract. So in a man, you have exosomes from the prostate, and in general case, you have exosomes from the bladder and the kidney, and very little contribution from the bloodstream. In plasma, you have systemic changes, and in cerebrospinal fluid, you have enriched material from the brain. What's very exciting with all this material that you can get from these biofluids is exemplified here, where we were asked to identify responders versus non-responders in a clinical trial where they were using an EGFR inhibitor. And typically when you sequence these, we saw over 2,800 total variants on the RNA. And if you zoom into the ones that are present in ClinVar, we found about 200 mutations that was represented in ClinVar. And here we also see this missense mutation in FCGR3A, which has been previously shown to reduce progression-free survival in colorectal cancer when you treat with an EGFR inhibitor. So this was actually a marker that we found to be verified in a different type of cancer. So it tells you that this method is really useful for both biomarker discovery as well as verification of known variants that affect treatment outcome. You can also use exosomes to boost your sensitivity for mutation detection. And here we exemplify that with a mutation called EGFR T790M. This is a mutation that caused resistance to first-line EGFR inhibitor therapy. So over the years, we've shown in 4 different peer-reviewed papers that when you use the exosomal RNA platform, you always get a benefit from the exosomal RNA in conjunction to the cell-free DNA. So if you're interested in studying mutations from a variety of cancer patients, doesn't matter almost what type of cancer, we've always shown that adding the exosomal RNA in your studies will add copies of your mutations and will increase your sensitivity. So we developed this EGFR T790M assay, and when we compared the performance of this assay to standardized assays like the Roche Cobas assay, which is the FDA-approved test for EGFR mutations, we shown that we can actually drive up the sensitivity from 58% to 92% and remain high specificity. So driving up the T790M specificity from 80% to 89% using the exosomal RNA. So that's quite exciting. Here's another example of a case report where a patient was found to have an EGFR positive tumor. They go on first-line TKI therapy, and in many cases, these patients become resistant to that therapy through this EGFR T790M mutation. Cell-free DNA assays often gives you false negatives when you're doing that, because some patients have very few copies of the circulating mutation on cell-free DNA. But when we combine this with exosomal RNA, many of these patients become positive, and that's verified in tissue, so the patient can start on active treatment that the patient can benefit from. And we've shown that with the comparison, about over 30% more patients would be eligible for the most effective treatment compared to the cell-free DNA assays if you utilize that approach. Another example is the EPI test, which is a commercially available urine-based liquid biopsy test. So this uses exosomes from urine. This is a test that has been commercialized now for a few years and has been helping over 50,000 patients in their prostate cancer management. So when you look at prostate cancer, it's a very common cancer in men, and the current standard of care is unfortunately fraught with a lot of risk and overtreatment and cost, because PSA that is currently used for screening has a very poor sensitivity and specificity. So the fact that urologists and patients need a new type of test to assess who should go in and do a biopsy is very evident. So we've sort of done a comprehensive clinical validation of this EPI test in over 5,000 patients from over 30 academic and community urology clinics in the US. The first validation of this test was published in 2016. We further did a second validation of this test in 2018, and a clinical utility study published in PCAN earlier this year. And these studies have all shown the efficiency and how this test can actually improve on the outcome for prostate cancer patients. As I mentioned before, exosomes in urine can be used for a variety of things, because exosomes in urine are coming from the kidney, from the bladder, and if the patient is a male, also from the prostate. However, the prostate exosomes are not feeding into the bladder, so the exosomes from prostates are only present in the first catch urine, the first void that comes out. So to standardize that collection, we have this prostate-rich first catch collection device where you urinate into this funnel, and the urine goes down here into this vial here. And the urine fills up, and the green thing is a float that goes up, and whenever the first 20 milliliters of urine has been collected, the remaining urine goes out this way into the toilet. And that's how you get a very well standardized first catch urine sample for prostate cancer evaluation. So it's a very simple test. It's a urine test. You pee in a cup, send in the sample to Exosome Diagnostics, where the exosomes are isolated. We crack the exosomes open, and as I mentioned, we find over 30,000 RNA targets in there. However, for this test, we only evaluate 3 by qPCR. That goes into a score that falls in on a range from 0 to 100, where the higher your score, the higher likelihood of finding high-grade prostate cancer on biopsy. And you get your result on a report that is very easy to interpret. But more importantly, from the performance perspective, the sensitivity is 92% with a negative predictive value of 91%. And this is really the important metric because that is how the test is used today. Specificity is not optimized for this test because that's not what the urologist is needing, because these men are ruled in for a biopsy already using the PSA, and this test would rule out unnecessary biopsies using its high negative predictive value. And this is we've analyzed thousands of patients on this, but of the 1,700 patients analyzed in the validation study, we can actually plot a very nice map over your risk score. So as you can see here, looking at the red line, which is your chance of finding high-grade prostate cancer upon a biopsy. If you have an EPI score around 30, you have around 30% chance of finding high-grade prostate cancer on a biopsy. If you have an EPI score of 50, you have about 50% chance of finding high-grade prostate cancer upon a biopsy. But you have about 80% chance of having any prostate cancer, including Gleason 6. We've also shown that this test, when used in a real-world scenario, we've shown that it actually improves standard of care, and doctors that use the EPI test finds 30% more high-grade prostate cancer compared to a blinded control arm that only used the standard of care for prostate cancer treatments. So exosomes are really, really important in management of these cancer patients. So this EPI test was the world's first exosome-based liquid biopsy, but there's many more to come. And it adds new information independent of known clinical variables in the PSA, for example. And it outperforms current standard of care and is included in the NCCN guidelines for prostate cancer early detection. So to summarize the exosome platform, we think that this is really a liquid biopsy application that goes even beyond oncology. It can be used to increase your sensitivity and your specificity for your biomarker, but it can also be used to develop multi-analyte tests. So as I mentioned, the exosome is packaged with RNA as well as protein and sugars and other things. So all of these multi-analytes can be used in your signature. And also, since you have exosomes being released from a variety of cell types, it goes beyond cancer, where liquid biopsies is not just for cancer, but also for example, neurological diseases, cardiovascular and infectious diseases. We have found that the exosomes can be very useful. And with that, I want to acknowledge some of the people that have been instrumental for the work that has been done here. So all the protein simple data on the JES was done in Munich, Germany, spearheaded by Lisa Meyer here. We also have support from, of course, the Protein Simple team and the entire Exosome Diagnostics Waltham facility as well. And with that, I'll take any questions.
Thank you both for your presentations. As a reminder to our audience, you may ask questions at any time using the question box. The audience has already submitted several questions, so let's get to them. Dr. Heger, Have any exosome researchers published using Simple Western yet?
Yeah, hi, hope you can still hear me.
Yep, we can hear you.
Yes, actually, yeah, great. So, yeah, as you saw, I listed that there's over 900 papers and patents using Simple Western, and I actually did look prior to the talk, and there are several that have looked at exosomes using the platform. And those include largely looking for specific targets of interest or to positively identify exosomes, and a little bit on the therapeutic analysis of them. Yep.
Dr. Skog, what is the difference between exosome RNA and circulating free RNA?
That's a great question. So there are many scientists out there that are trying to get RNA from total plasma or other biofluids, where they just take the entire sample and precipitate everything using caltropic salt or other methods, and they isolate all of the RNA in the sample. What we've seen is that this is actually a suboptimal approach for most applications because it blurs your signal. So there's RNA components in that sample coming from a variety of sources, including microRNAs in AGO2 proteins, you have HDL, you have even platelet material that all sort of confound your data. And it's important to point out here that more is not always better, right? So a lot of scientists are trying to figure out ways to get more material, more RNA or more protein out of the sample. But if that RNA or that protein is not coming from the disease process, more is not going to be better. It's just going to blur your signal. That's what we've seen in many cases for the CF RNA or cell-free RNA, where you get a more cleaner prep from the exosomal material. So it increases your specificity for a variety of markers.
Thank you. Dr. Heger, what is the difference between Western blot and capillary electrophoresis?
Sure. So the classical Western blot, you're transferring your proteins after they've been separated on an SDS page gel to essentially a piece of paper called the membrane. So you have an orthogonal push and catch with kind of a transient catching of the proteins on that piece of paper. Now, capillary electrophoresis, as used in Simple Western, essentially takes that entire process and puts it in a tube. You can think of a capillary just as a small tube, and we performed the separation as well as the entire immunoassay in there. So you're not left with all those individual steps and variations and movements from different setups. It's all contained and performed within the same exact capillary. That gives you a lot more reproducibility, consistency, and so forth, as I mentioned. Thanks.
Thank you. Dr. Skog, what is the RNA input required for next generation sequencing?
Another great question. So we've shown that you can do complete RNA profiling down to 0.5 ml of plasma. So, and about a ml, between 1 and 5 ml of urine. So it's a very powerful approach. That the smaller the volume of biofluid that you have as input, the less RNA you will get. So it's on that. If you do your isolation properly, it's all linear. So you double the volume input to your assay, you will see twice as much RNA. So since different RNA molecules are present in different amounts, it's very important to know what marker you're looking for. So more is always, so if you double your volume, you see more of these low copy number RNAs, and those will fall out the smaller volume you go. So there's no sort of minimal required amount because it depends on the marker. If you go down to 1 microliter of plasma, for example, you'll only see the most abundant RNAs. like ribosomal RNAs, tRNAs, and things that may not be useful as a biomarker. So it's important that you address the question of minimal volume to what marker you're actually looking at, because since there's a very wide dynamic range in the copy numbers of different types of RNAs.
Thank you. That's interesting. Dr. Skog, another one for you. How do you assess the quality of the RNA before next generation sequencing? Are there any criteria to assess your sample at library prep?
Yeah, so we have our internal quality control that we always perform before the NGS, and I think I don't want to say that there's a standard way that would work for every application, because the QC process that you utilize for your library prep depends on the marker that you're actually interested in looking at. So for a more abundant marker, your QC prep may be a little bit more laxed than for a marker that is down in the single copy number region. So at Exosome Diagnostic, we have our standard sort of way of QCing that, but I think every lab that develops their own RNA sequencing method needs to assess the QC metric depending on what they're looking at.
Thank you. Dr. Heger, you mentioned the PEGI-SU. With the PEGI-SU in particular, you can probe multiple times. Is there a risk of cross-contamination?
Yeah, so it's a good question. So I didn't get a chance to show how the PEGI-SU functionally operates, moves between capillaries and so forth, but no, there isn't any risk of cross-contamination. A new capillary is used for each of those individual probings, and the elegance and the beauty of how the machine is built and operates ensures that there's no cross-contamination.
Thank you. Dr. Skog, does proteins or nucleic acid have higher potential as biomarkers, in your opinion?
That's a great question. So I think both of them have great potential as biomarkers. When you look at RNA, the advantage of RNA is that you have very simple analytical platforms for the whole diversity of RNA targets, right? So with RNA sequencing, you can easily sample and analyze over 30,000 different types of RNA, and you can monitor the amounts, you can look at splice variants, you can look at SNPs, etc., using NGS, a single platform, which is beneficial. When it comes to protein, it's been challenging to have that kind of a multi-analyte sort of spectrum. You can look at up to 1,000 proteins or so, but these multi-analyte methods haven't been easy to scale. So, but that being said, I think the biomarker potential in RNA and protein they're both very, very powerful. So it depends on the application.
Thank you. Dr. Heger, for exosome marker characterization, if I'm seeing very high background in my Western blot, am I going to see that in a simple Western?
Yeah, so a high background in any immunoassay is largely driven by the antibody itself. So typically, if you have an antibody with high background, you may, but not always, see that on simple Western. The antibody incubation is done quite differently, and we do have a special antibody diluent we use to help with that as well. But generally speaking, if you have a high background antibody, there's a chance that you'll see that too.
Thank you. Dr. Skog, did you perform proteinase K or DNase treatment, and would you say that that's needed before next generation sequencing?
Yeah, that's a great question. If you're interested in only looking at the RNA, so RNA levels, you have to ensure that your method does not co-purify DNA. So that can be done in a variety of different ways, and you can control for it in a variety of different ways. It's important to know that if you have small amounts of RNA, even single genome equivalents on the DNA level will significantly affect your result, because the genome is so large and you can get reads from a variety of different regions. So if your analysis is RNA levels only, I would make sure that you address that point.
Thank you, Dr. Skog. And now we have time for just one more question, and that question would go to you, Dr. Skog. Would you say that exosomal information provides better information on biomarkers, or would you say that using this data from exosomes should be combined with other techniques, and that would provide better information on biomarkers?
Yeah, I've been sort of a strong proponent of that for many years now, where we showed that for some diseases, you need to use all your tools in your toolbox to have an efficient biomarker, and especially going into like early detection of cancer, I certainly think that combinations is very important. And the strength here with exosomes is that you can use combinations of proteins, RNA, DNA. You can even combine it with cell-free DNA if you want to. So absolutely, I think a multi-analyte approach is going to be beneficial.
Thank you. Unfortunately, that's all the time we have for today. If you have any further questions, please consider reaching out to the Protein Simple team directly. Their email is shown on the screen. As a reminder, the webinar will be archived on the scientist website, and you'll receive an email notifying you when the on-demand webinar is available. I would like to thank everyone who took the time to join us today, and particularly those of you who shared your questions and comments. On behalf of the scientists, I'd also like to thank our speakers, Dr. Chris Heger and Dr. Johan Skog, as well as our sponsors, Protein Simple and Exosome Diagnostics. Thank you, everyone, and have a great rest of your day. Goodbye.